So hello everyone. My name is Manjot Nagyal. I'm the USABO club coordinator at Center for Excellence in Education. I'm really excited to introduce you all to our past USABO alum, Catherine Wang, who is currently a MD PhD candidate at Johns Hopkins University. And from here, we'll go ahead and get started.
Great. Thanks so much for having me. Hi everyone. My name is Catherine. Like she said, am a second year MD PhD student at Johns Hopkins. And I'll be talking about my experience in research in medicine throughout high school, undergrad, and my current graduate program so far. I know that this talk is affiliated with USABO, and I will talk a little bit about that, but the point of this particular presentation is really to discuss the academic path I've taken a little more broadly, not just USABO study strategies, which I've actually given another talk about. But yeah, so I really want to focus on kind of how to find research opportunities in high school, and also in the long run, what are the different versions of graduate school, including, what the heck is an MD PhD program. And at the end, hopefully we'll have some time for questions if you have any. Before we begin, I do want to make you aware of some disclaimers. One is that I'm going to mention a lot of mentors that I worked with throughout high school and undergrad, and that is not necessarily an invitation for you guys to reach out to them. I didn't actually get their permission as to whether or not they want high schoolers reaching out to them. And so this is not an invitation to do so. And also, some of the research and shadowing experiences I had as a high school student may have different restrictions around them now. So an example would be that I actually shadowed in an operating room and saw a surgery when I was in high school, but when I tried to do it again in college, they actually wouldn't let me. So the rules are always changing. And of course, this is meant to be kind of an informative example rather than a template of how to move through this field. So I want to give you guys some context about just who I am and where I grew up. I'm from Lexington, Massachusetts. This is a very historical town. It's known because it's where the Revolutionary War started. And it's kind of on the outskirts of Boston. You can see I've labeled it with a little heart. And so it's around like 20 to 30 minutes into the city. And that ends up being very important. Because there's a lot of universities in Boston. But nowadays, Lexington's really known for its excellent public school system. And I was fortunate to be able to have access to that as a middle schooler and high schooler. But I want to dispel the myth that I was always very interested in biology and very competitive in the STEM fields. Up until high school, I lived a pretty carefree and non-academic life. I played soccer, I drew pictures, but the one thing was that like many kids, I was very fascinated by, nature and plants and birds and sponges. And I watched a lot of nature documentaries. Sometimes my parents, instead of taking me to summer camp or daycare, would just drop me in a bookstore and I would read these nature encyclopedias for fun. And so I always knew that I was very interested in living organisms and ecosystems, but all my friends were doing math team and science team, and I really wasn't. And so I kind of thought that I wasn't particularly cut out for science competitions. I thought I didn't have the personality for it. But when I got to high school, I figured I had to pick something to focus on, and I wanted to keep learning biology because I thought it was interesting. And at the time, the most competitive science team at Lexington High School was the Department of Energy National Science Bowl. And for those of you who aren't familiar with this competition, it involves teams of students who answer science questions by buzzing in, and people on the same team tend to specialize in different topics. So I joined this competition and eventually became kind of the token biology person, which was actually very hard to do because this competition is really kind of advanced. They make you learn a lot of things. And these are pics of our high school A team throughout my 4 years of school. And you'll notice that in 3 out of the 4 years, I was the only girl, which was its own challenge. And I want to reassure everyone that the fields of biology and chemistry and kind of just STEM in general are not actually as kind of male dominated as they seem to be at the high school level. I'm not really sure why they're so male dominated, but that was certainly very intimidating, but it was also very motivating because I felt like I really had to prove myself as the only girl on the team. And so that kind of lit a fire in me to really give it my best shot. And the other benefit of doing Science Bowl was that it exposed me to a lot of upperclassmen, and their advice really helped because they're further along than I was. They're more familiar with kind of the high school and like the structure of these competitions than I was. And I could just follow their example, but kind of have, you know, a 2-year head start even. So of course, I also did the USABO, which is why I'm here. I have to say that the USABO is probably the hardest thing I've ever done in my entire life. So for people studying for USABO, you can be reassured that it actually gets a little bit easier, I think, after USABO. But the kind of combination of USABO and Science Bowl really motivated me to independently study biology at the college level for 4 years in high school. And, you know, the crazy thing about these competitions is that they expect you to magically learn this content and kind of apply it under crushing time pressure, which is a really tall ask. And to prepare, I ended up reading a good chunk of all of the books I've put up here, and also a lot of Wikipedia. And even though this was a lot of hard work, I think that the foundation of this knowledge was very empowering in the long run. And it was also very enjoyable to collect. I think learning all this for the first time was actually really intoxicating experience. It was like having a candy box and getting to like sort through all the candy, except you have to also memorize the candy. But the thing about these competitions is that they really force you to become a generalist, which is really rare even at the undergrad level. And so you're able to appreciate biological phenomena sort of at all length scales. And having such a honestly advanced background at such a young age enables you to discuss science at a level that like other kids your age can't. And at the end of the day, that kind of made me brave enough to talk to professors and kind of professional scientists, even though we were not equals in terms of knowledge. Obviously, I at least could ask some kind of half-formed questions. So that knowledge base really came in handy when I decided that I wanted to do research. And the reason I even started thinking about doing research at all was because my upperclassmen Science Bowl friends had been doing it over the summer. And I was like, how do I do that? Like, how do I magically end up in a lab? And so the way research normally works in high school is there's 2 ways you can work in a lab. One is through kind of a structured program, and they normally happen over the summer. Some of them you have to pay for and some of them don't. And generally the ones that you don't have to pay for are very competitive to apply into. Or, you know, if you're too young to qualify for those programs or they're, you know, out of budget, you can always just email some nearby professors and hope that they let you into their labs. And so that's what I ended up doing. I first started looking for research opportunities when I was like, I think in the fall of my sophomore year of high school. And this was because we had a science fair coming up and I was required to participate in it because I was taking AP biology. And I wanted to actually do a project in a lab instead of doing it, you know, in my kitchen. So yeah, I decided I wanted to find a lab. And I was conveniently located close to Boston. And I thought this would meant that I would have tons of options. I mean, Boston is home to Tufts, like BU, Harvard, MIT, Mass General Hospital, Dana-Farber Cancer Center. The list is very long. And so I thought I would have some really difficult decisions to make once I started emailing people. But that was actually not the case at all. I remember sending out just a handful of emails, not hearing back from anyone. I started sending out more, just completely at random, nothing. So here is an example of an email that I sent in 2015. I would often, if I did get a response that looked like this, like, I really wish I could accommodate you in our lab, but because we work with many toxic drugs and animals and radioisotopes, it'll become very difficult to have a high school student working here. And yeah, so it's actually, I didn't realize it at the time, but it's a big ask to have a high school student working in a lab. The professor takes on a lot of liability, and you take up the time of everyone who has to mentor you because you don't actually know anything. And so what that means, though, is that if you send out enough emails, the people who do say yes are actually very invested in your progress. And so I think for my first research position, I must have sent out maybe 30 or 40 emails. And I think I got maybe 5 no's. and one yes. And once I got a yes, I just stopped. And my first yes was from this professor at Boston University. Her name is Kimberly McCall, and her lab studies the molecular mechanisms of apoptosis, which is a form of programmed cell death. And so the model that she uses is the fruit fly ovary, which is shown here. And so basically the entire abdomen of the female fruit fly is made out of these ovaries, and they're made up of chains of developing eggs surrounded by these nurse cells. And if you dissect out the ovaries and then stain them with a DNA dye, you can actually visualize the process of apoptosis. And because these are in fruit flies, you have a whole suite of genetic tools to use to create knockout lines and things like that. And so you can kind of correlate the function of a gene by knocking it out in a fruit fly and then looking at the effects on the rate of apoptosis, which is very cool. So this was my first research project ever. It was for the school science fair. And it was called The Effect of Over-the-Counter Drugs on Cell Death in Drosophila Ovarian Tissue. It was the first experiment I ever designed. I did it independently. And basically what I did was I went to CVS, I bought drugs, I ground them up, and I fed them to flies when they were larvae. And the flies were from different genetic backgrounds. And then after 2 weeks, I would harvest their ovaries and then count the percent of apoptotic follicles. And then, make some cute little bar graphs to say that like, oh, when I feed ginger to fruit flies of this genetic background, the level of apoptosis, changes in this way. So obviously there are many issues with the experimental design here, but it was a good start and I really enjoyed my time there. The lab members were all really nice to me and really took the time to explain what they were doing and actually taught me some like really valuable microdissection and imaging skills. One interesting phenomenon that I saw was that when we fed wild type flies Tylenol, their nuclei tended to kind of cluster together in pairs like this. And so I'm pretty sure that someone else from the lab after I left actually tried to follow up on this to see if there was something going on with like the spindle. But anyway, that kind of, was my first introduction to research. I caught the bug. I wanted to do it again. And so that was kind of the beginning of everything. So from this experience, I also started to get an idea for how biology labs are organized in general. And I wanted to take this opportunity to walk you guys through this because everyone kind of seems to assume that this is common knowledge. And maybe it is if your parents are, from biological backgrounds, but that's obviously not the case for everyone. It wasn't the case for me. And so I feel like I would have appreciated someone actually explaining this to me. So, anatomy of a biology lab. You have your PI. This is your primary investigator, and they're the head of the lab. And so the lab will be named after them. And they are the people who kind of run the show. And importantly, they apply for grants. And so they kind of devise the kind of research questions that lab is going to ask, writes it up and submits it to the government for money. And so without the PI, there is no money. And so they kind of have the final say in what goes on in the lab. Each lab normally will have some PhD students. These are graduate students in specifically PhD programs. And so they can expect to take between 4 to usually 6 years in one lab to kind of create this project and see it through. And so usually early PhD students are still learning the ropes, but by the time, they're 5 or 6 years in, they're really, really knowledgeable. And so if you're a high school student, often you'll be working with a PhD student as your mentor. You'll also see some rotation students. These are graduate students who are in the very beginning of their PhD program, and they'll be kind of joining labs for 1 to 3 months at a time to just try it out. So they're not actually part of the lab, they're just there for a couple months. And so they'll often be there over the summer. You also have postdoctoral researchers. These are scientists who have already completed their PhD and are doing their next phase of training. And so they tend to be super knowledgeable, and often they bring to the lab a new skill set that they developed during their PhD that they're applying towards a problem that the lab is particularly good at solving. And so they also tend to be a little older. And there are also staff scientists and lab techs, and these are non-trainees, so they're just normal employees, and they provide technical support for the lab. And sometimes a staff scientist can be with the lab for like 20 years at a time, so they'll often be the most knowledgeable people technically. And then of course there are undergrad trainees, and sometimes there are even high school students. And so usually undergrads or high school students will kind of sometimes they'll join the lab for a long time. Sometimes they won't join the lab for a long time. It'll just be a couple months. But generally, this is an incomplete but overview of kind of the hierarchy of a normal biology lab. And the kind of axes of variability of a biology lab are as follows. The PI can be very hands-on or hands-off. Hands-on would mean that they're really involved in the day-to-day proceedings of the lab. They're kind of maybe hovering over your shoulder and like holding your hand through experiments, which can be really helpful, especially in the beginning. Or you can have a really hands-off PI that kind of lets you do whatever you want. And the downside of that could be that you don't actually get that much direct guidance from them, but also you have more intellectual freedom. PIs can also generally be like very new faculty or more established faculty. And new faculty tend to have smaller labs, and so they can afford to be more hands-on. And they also tend to have less funding than very established labs, which are bigger and have more funding, but again, could have a more hands-off PI. You can also kind of have different demographics within the lab. So some labs are really grad student heavy and some labs are very postdoc heavy. And generally, I think that the training environment tends to be a bit more like learning focused when it's grad students, because the grad students are there to pick up new skills, whereas postdocs tend to be more productivity oriented. But obviously, there are exceptions to this rule. And lastly, the nature of research in a biology lab can obviously be different. And people often will kind of think about it along a spectrum of basic to translational to clinical, where basic research is really focusing on the kind of fundamental mechanisms of biological phenomena. So like, how does this gene get regulated at the molecular level? Whereas the translational lab would be kind of looking at therapeutic opportunities, but doing so in mice. So it would be like, how does this cancer treatment, or does this cancer treatment help mice with pancreatic cancer? And if, you know, the translational results are promising, you might even end up with a clinical study where you'd actually be seeing how like new drugs or medical interventions are doing in patient populations. And so usually labs will not occupy more than one of these areas, although again, there are exceptions. And so the kind of flavor of the research really depends on which like area of the spectrum you occupy. Okay, so back to the research journey. So after my time in the McCall lab, that was a fruit fly lab, I joined the Poznansky lab at Mass General. And this was run by Mark Poznansky, who is an MD PhD, actually. He's an infectious disease doctor and also an immunologist. And the reason I joined this lab in particular was because I had gone to some talks in Boston over the school year that happened to be about immunology. And I became really interested in how the immune system can be leveraged to solve clinical problems that way. And so this lab in particular was interested in type 1 diabetes. And the way type 1 diabetes works is it is an autoimmune disorder, and it causes the elimination of your insulin-producing pancreatic islet cells. And so when you lose those cells, you lose the ability to produce insulin, and you also lose the ability to regulate your blood sugar. And so it's a pretty devastating disease, but nowadays people treat it with insulin. So they had this idea that they wanted to, instead of injecting insulin every day as a treatment, they wanted to actually take pancreatic islet cells from a healthy individual and eventually transplant them into a diabetic individual such that they could have their own source of insulin. But the issue there is that when you do these kind of transplantations, your immune system recognizes the transplanted tissue as a foreign object and launches an immune attack against it and basically degrades it. And so the kind of project that was going on in the lab at the time was to figure out how we can take these pancreatic islet cells from mice and then encapsulate them in this kind of gel matrix. And we would put chemicals in this gel matrix that would specifically repel immune cells like white blood cells and prevent them from attacking the kind of islet cells that are hiding inside this matrix. And so you would encapsulate these matrices, or you would encapsulate these cells in the matrix, and then you would inject them into a diabetic mouse, and then you would try to see if the diabetic mouse is able to control its blood sugar, and then later you would actually take these cells back out of the mouse and see if they had been damaged by the immune response. So my job in this lab was really just to like take pictures of these islets that had been taken out of the mouse and then see to what extent they had been damaged by the immune system. So yeah, that was a cool experience. And while I was at MGH though, I had this idea that I should shadow in the hospital. And I came to this idea because I had seen on College Confidential actually that other kids my age somehow were able to use their like parental connections, if their parents were doctors, to go witness these surgeries. And my parents aren't doctors, but I figured if I can email my way into a lab, I can probably also email my way into an operating room, an OR. And so I emailed a bunch of people at Mass General. And to my surprise, someone actually got back to me within 5 minutes. And his name is Alum Goldstein. There he is right here. And he's a pediatric surgeon. He's actually the chief of pediatric surgery at Mass Gen. And he specifically treats patients with congenital malformations of the GI tract. And he also runs a lab studying the same diseases that he treats. And so this was really mind-blowing for me because through kind of talking with him and shadowing him, I got to talk to the family of a patient with this disease called Hirschsprung's disease, where they kind of don't have a motile large intestine. And then I got to see the actual surgery where they repaired it. And then later I actually got to join in on one of his lab meetings where I got to see them discussing fundamental research about, why this happens at all. And so that was my first exposure to the idea of a physician scientist actually at work, like this bench to bedside translation of applying science to medicine. And so that's kind of how I became interested in the idea of an MD PhD, which I'll talk about later, and just the duality of science and medicine. Until now, I had sort of thought that they were completely separate and that you had to do one or the other. So my final high school research experience was actually through a program. So no more desperate emailing, just an application. I applied to this program called the Broad Summer Scholars Program. It's held at the Broad Institute, which is this massive genomics research institute that's kind of run by MIT and Harvard. And this is actually where the Human Genome Project was done. And so it's a famous and well-funded place. And the way this program worked, and you can actually still apply to this, they take around 10 high school seniors from the greater Boston area, and they pair you with a mentor in a lab at the Broad Institute, and you kind of do a 3-month project with them. So I was paired with a research scientist from the Pan lab. So this is the PI, her name is Jen Pan. And the project was very interesting. Basically, at the Broad, because they have so much money and sequencing power, they have the ability to do what's called a genome-wide association study. And basically the way this works is that they can take hundreds of patients that have the same disease and they can do whole genome sequencing on them. And they can kind of correlate like which sites in the genome tend to have higher mutation rates in people who have the disease versus people who don't. And so like what this plot is showing is the x-axis, you have chromosome number. So this is genome location, and then sort of the relative frequency of mutations at each of these locations. And everything that's above this significance threshold is kind of considered an interesting target that might actually have a real correlation with this disease. And GWAS is a really great way of kind of trying to figure out potential therapeutic targets or just biological mechanisms behind diseases that you don't really know what's going on. And so one such disease, and the disease for which this GWAS plot was actually created for, is schizophrenia, which some of you might be familiar with. But this is a disease where people have a lot of auditory hallucinations, and but people don't really know exactly what's going on a molecular level. And so one of the peaks from the schizophrenia genome-wide association study was this calcium channel called CACNA1I. And that's kind of interesting because this calcium channel is expressed by neurons, and schizophrenia is a psychiatric disease. And so we wanted to know how, very broadly, mutations in this ion channel would actually cause, dysfunction at the neuronal level. And so we wanted in particular to see how these patient-derived mutations would actually affect the ion channel function, and in particular its subcellular localization. So for example, if you had this like this particular mutation here, the question was, would that potentially prevent the ion channel from being able to traffic successfully to the plasma membrane? And so my particular project was to create a pretty basic membrane localization assay for these patient variants. And so basically I took a red fluorescent protein and I stuck what's called a farnesyl group onto it, and that causes it to insert into the membrane. And then I tagged this channel with a green fluorescent protein, and then you could kind of see areas of overlap between the membrane and the GFP. And so the extent to which, you see this green signal co-localizing with this red signal is how much of that protein is actually being trafficked to the membrane. And so after I left, they in theory could use this assay to start looking at the different patient variant. Okay, so that was the completion of my high school research. And these were some of my takeaways. One was how to use public transportation. Another was how important good mentors are, obviously. Only a good mentor would even let me in. But also, more broadly, I learned that even when you don't know anything, it means a lot to just show up. These professors don't actually expect a high school student to know very much. And so you don't actually need to impress them with your knowledge or anything. You just have to show up and be enthusiastic. And you'll find that, especially at that age where no one has any expectations for you, any kind of question you come up with or any kind of enthusiasm you can demonstrate for science is actually really respected and taken seriously by the people around you. So I wouldn't be held back by any notion that you're too young to be in a lab. It just takes a lot of emailing. Okay, so kind of the next chapter of all this was undergrad. And I decided to go to Stanford for undergrad because I wanted to go to California. I had grown up in New England and I was very curious about sort of what the academic scene looked like on the West Coast. So I majored in biology and minored in art practice. And for the first year of undergrad, want everyone to know that I was completely burnt out from high school. My senior year of high school was very successful. I competed at the International Biology Olympiad. I did Science Bowl again, and we actually won that year. But what that meant was that I was studying as intensely as I had for all of my high school experience during my senior spring and summer when all of my friends were really taking it easy and going on vacation. So this is actually a picture of me asleep on a textbook in a Starbucks that one of my friends took through the window. And I was just so exhausted. And so for the first year of undergrad, I didn't do anything. I didn't join any labs, didn't do any extracurriculars. I just kind of partied and took classes and slept. And it was very much needed. But the one thing I did do was I stayed involved in Science Bowl for nostalgic reasons. And that actually led to an interesting research opportunity. While I was at the Bay Area Regional for Science Bowl, I ran into this guy, Rishi Kulkarni, who I had actually known for a couple years because he's very involved in Science Bowl. He writes most of the questions. And so we started talking while we were volunteering together. And at the time, he was a PhD student at Berkeley, and he was moving to Stanford for a postdoc the next fall. This was freshman spring of college. And so he was just like, you should work for me. I need an undergrad to help me. So like, why not? Why don't you just do it? And I said I needed to think about it. And he told me that he would be working in Carolyn Bertozzi's lab. And when I did some Googling of who Carolyn was, I found out that Carolyn actually also grew up in Lexington and went to LHS. And so without learning any more about the science, I was like, okay, I will just join this lab, I guess, because it seems like the universe just wants me to. Which actually, in retrospect, was a very good move because I knew that the people I would be working with were solid. I had known them, and Carolyn had an amazing reputation. And so I kind of thought that, no matter what the science is, I'll probably find a way to be interested in it. The wild card is actually the people you work with. And so that was the basis by which I joined this lab. And, if the name Carolyn Bertozzi is familiar to you, it's probably because she won the Nobel Prize in chemistry this past October, which was a big deal. And we all sort of saw it coming, but we didn't know when it would happen. And I really wish that I was there when it happened, but that's okay. But anyway, this is an example of a really, really big hands-off lab. So Carolyn was more of kind of our inspiration than a daily hands-on mentor. And so everyone in the lab was very capable and very independent. And Rishi and I basically had complete freedom to do whatever we wanted in that lab. And so, yeah, the Bertozzi group in general is famous because it invented bioorthogonal chemistry, which is what Carolyn won the Nobel for. But this type of chemistry, which can be done in living organisms, was used to revolutionize the field of glycobiology, which is the study of glycans and their contribution to biological phenomena. So glycans, which are shown here, are these chains of sugars. We normally think of sugars as just kind of food, fuel for the cell, but glycans can actually kind of elaborate into these really fancy structures that decorate the outside of the cell, and they form this layer of sugar coating called the glycocalyx. And Rishi and I in particular were interested in the neuronal glycocalyx, so the glycocalyx surrounding neurons. And this is a component of neurons that's frequently ignored, mostly because the glycocalyx is hard to study. That's why a new field of chemistry had to be invented in order to study it. But it's very important because the physical and chemical properties of the outside of the neuron are almost completely dictated by this giant layer of sugar. We always say that, the topology of the cell is more like this chocolate-covered almond than anything else, where this chocolate is the glycocalyx. And the neuronal glycocalyx is particularly weird because it has a high concentration of this little purple thing over here, which is called sialic acid. And it's this charged sugar that tends to coat the very ends of the glycans. And one interesting thing is that, neurons have this whole other axis of activity to them. Normal cells, you can study like what proteins they express and what's going on inside of them. But neurons have this whole other dimension where they have electrical activity. And the contribution of the glycocalyx to the electrical activity or the electrophysiology of neurons was very poorly understood. And so we were curious about that, partially because it had been shown that changes in the sialation state of neurons were strongly associated with disease states. And so when they looked at post-mortem brains of patients who had, Alzheimer's disease, schizophrenia, or prion disease, you could see that the sialation patterns were really different from normal. But while these were some really strong correlations, we didn't actually know, and no one knew if there was any causative relationship between, aberrant sialation and the kind of messed up neuronal activity that occurs in these disease states. And so to kind of look at this in an isolated system, we first artificially removed the sialic acids off of neurons with a bacterial sialidase. So we were culturing neurons in a dish. We added a bacterial sialidase, which would go and just cleave off all these sialic acids. And then we used voltage recording techniques to see how the activity of the neurons actually changed. And to sort of make a long story short, we found that removing the sialic acids caused the activity of networks of neurons to become less connected. And so their activity became sort of less correlated with each other. It was more random than synchronous, which is usually kind of a bad sign. So yeah, that was actually some of the first evidence that the glycocalyx actually affects neuronal activity at the network level. But anyway, while that's cool, we wanted to see if a similar phenomenon can actually happen under physiologic conditions, i.e. is this phenomenon relevant in a real brain rather than just a dish of cells that you've added a bacterial protein to, because that would never happen in real life. So because we're interested in neurological diseases, we honed in on this cell type called microglia, which are the resident phagocytes of the brain. And so normally they kind of help keep neurons healthy. They remove kind of trash and random little vesicles in the brain and keep everything clean. But under disease states, they become chronically what's called activated. And then they are a hallmark of neurodegenerative disease, and they really drive neuroinflammation. They're responsible for the neuronal damage that occurs as a result of neuroinflammation. And interestingly, something we found in the literature was that microglia, when they're activated, secrete a sialidase, and that sialidase had not been identified. But we thought that everything was kind of matching up, and in accordance with the previous results that desylation could disrupt neuronal activity, we hypothesized that this sialidase might have a yet undiscovered role in modulating neuronal activity. And that would sort of draw a nice link between inflammation, which involves, activated microglia, and disrupted neuronal activity, which we know happens in disease states. So again, to make a long story short, we ended up characterizing this really interesting pathway where first we identified the mystery sialidase from activated microglia to be a protein called neuraminidase 3. The name doesn't matter, but we found that it was actually secreted in exovesicles, which are these tiny little membrane-bound structures. And the enzyme was embedded within the membrane. And when you treat neurons with this stuff, the exovesicles actually fuse with the neuronal membrane and deposit this enzyme within the membrane, within the neuronal membrane itself. And then from that position, the enzyme is able to desilylate the neuronal glycocalyx. And when we looked at how the neuronal activity was affected, we saw that the connectivity of the network went down overall, which was consistent with our previous results. So this was actually the first time we could present glycocalyx remodeling as a mechanism by which inflammation could lead to disrupted neural activity, which has some implications for how neuroinflammatory diseases are thought about and presents this neuraminidase 3 potentially as a therapeutic target. So that was my undergrad thesis. And around when the pandemic was hitting was when it was time to apply to grad school. And the question was, what kind of grad school should I go to? It's not super common, although it's becoming more common to just go straight into a job after you major in biology. Typically, those jobs are at pharmaceutical companies and biotech companies. But if you want to actually be a scientist in an academic environment, you have to go to grad school. And so I was very torn between these three options. Did I want to be an MD like my good friend Alan Goldstein? And he actually also runs a lab, but he doesn't have a PhD. Did I want to be like Carolyn, or as, similar to Carolyn as humanly possible, who is a widely impactful and successful PhD scientist? She doesn't have an MD, but she does very impactful research and has started several companies that are leveraging therapeutics into clinical trials. Or do I want to be like Mark Poznansky, who is actually an MD PhD, and he runs a lab and sees patients? And so that was kind of an existential crisis for me. And so while we're here, I want to take the opportunity to kind of discuss the differences, because this is also something I had to figure out by myself, and I kind of wish someone would just tell me. So how does an MD work? So MD stands for medical doctorate. It's like what you go to med school for. You begin by doing 4 years of undergrad, and while you're an undergrad, you're a pre-med, so you have to fulfill these pre-med requirements. And typically that means taking these foundational STEM classes like biology, biochem, gen chem, orgo, physics, math. And if you want to be a competitive applicant, your grades have to be pretty good. And this is one reason why pre-meds tend to be very stressed in undergrad, because not many other career paths require you to have such a good GPA. Not even grad school. The big exam that you have to take as a pre-med is the MCAT. This is kind of like the SATs of med school. And the MCAT has different sections. There's a reading section, there's a physics chem section, there's a bio section. I have to say that the biology section is a bit easier than the USABO semifinal, actually. So if you're very well prepared for the USABO semifinal, the MCAT biology section should not be too much of a problem for you. And on top of all this, you're expected to have research experience in a lab, some clinical experience like volunteering or shadowing in the hospital, and whatever leadership means, but that's also a requirement. So again, this is why pre-meds are so stressed. They have to do a lot of things. After that, you apply to medical school, which is a pretty grueling process. People usually apply to between 15 to 35 medical schools, depending on, how wide you want to cast your net. And there's essays associated with each of them. It's very similar to the college application process, except there are interviews, which are very high stakes. After you presumably get into a med school, that usually will take 4 years. It's a full-time position, and med school costs money. So there is a pretty hefty price tag associated with going into med school, and a lot of people go into debt for it. There's also a good amount of financial aid in many cases, but that's never a guarantee depending on which institutions you get into. So regardless, it's expensive. After you go to med school, you will apply to and do residency. And the length of the residency is going to depend on what specialty you want to do. So for example, internal medicine is 3 years long, but neurosurgery is 7 years long. So it really depends on what your interests are at this point. And if you want to subspecialize after your residency, you will do fellowship. So for example, if you want to be a cardiologist, which is a heart doctor, you have to do 3 years of internal medicine residency, and then you have to do, I think it's 3 years of cardiology fellowship. So you're in training for a really long time. And only after that can you actually get a job as what we call an attending, seeing patients. And that's when you actually start earning a good amount of money. Until then, you are earning somewhat less than minimum wage if you count all the hours that you work. So training is very long and it pays off in the end, but it's very much a case of delayed gratification. And you can incorporate research into this path, but it usually will add extra years to your training. So for example, you can take an extra research year in medical school if you want to, but that just adds an extra year. You can do research during your fellowship. You can even get a PhD during your fellowship if you want, but that adds extra years. This is something that a lot of people do, but it will extend your training time. Okay, moving on to how a PhD works. So again, we have 4 years of undergrad and you have to fulfill graduate program requirements, but this is much looser than pre-med requirements. Generally, it's like take relevant coursework for the department you're going to apply into later. And they don't really care that you've covered specific courses. They just want to see that you've taken a rigorous and appropriate course load. And your GPA doesn't necessarily matter as much as, it did in med school, because they would rather you take a hard course and get a B than take an easy course and get an A. Whereas like pre-med admissions committees actually just want to see a bunch of A's. So grad school is a little different. You apply to PhD programs that also involves essays and interviews, but generally people apply to fewer PhD programs than med schools. And then you'll do your PhD, and this can take between 4 to 7 years. I've seen longer, but it really shouldn't take longer than 7 years. And this is when you receive your PhD, and you actually don't have to pay to do a PhD program. I didn't know that until like late undergrad, but you are given a stipend, and it's not a very big stipend, but it is a stipend. And typically you'll do several rotations in different labs before you choose the lab you're going to join. You usually do that by the end of your first year. And then in the meantime, you're also taking classes for the first year or two of grad school. Afterwards, once you stop taking classes, you work on your thesis full time, and you graduate when you've written a complete thesis. And again, graduation time will really depend on like your own goals, like how impactful do you want your PhD work to be, and also sort of what your PI envisions for you, and also how lucky did you get with picking a fast-paced project versus a project that had a lot of unexpected hurdles. After your PhD, you can choose to do a postdoc position. And, this will involve joining another lab and kind of doing a couple of years of research there. People normally do postdocs to just beef up their academic resumes. So if you actually want to be a PI later, you absolutely have to do a postdoc, because it's going to show that you can do research independently. But some people who go work at companies after their PhD also opt to do a postdoc because it kind of makes them more competitive on the job market. But lately, that hasn't been the case as much. Some like postdocs are becoming less and less popular, and there's actually a shortage of them right now. Anyway, and then after your postdoc, or after your PhD, depending on what you do, you can finally get a job. And the job is usually going to be either in academia, where you're part of a university and you're doing research, or in industry, where you're like working for like a pharma company or a biotech company. But also, if you have a PhD, you can do like a lot of kind of random things that you wouldn't even think about. Like some people go and work in like venture capital firms. So it's very random. But generally, the approach to your PhD is going to really depend on whether you want to be competitive for an academic position or an industry position. And if you want to compete for an academic position, then there's a lot of pressure to publish. And so being a PhD is actually really hard. It's very frustrating, but you come out, knowing how to do independent research, which is a very valuable skill set. Lastly, what is an MD PhD? I'm sorry, I know these slides are really dry and not very colorful, but hopefully this information is useful. Anyway, I'm an MD PhD. It works very similarly to kind of a combination of MD and PhD with some kind of changes. So you have to do 4 years of undergrad. during undergrad, you have to fulfill pre-med requirements and demonstrate, quote, exceptional research capability. Although I think they tend to care more about research and less about clinical experiences here. You have to apply to MD PhD programs. There are fewer MD PhD programs than med schools out there. And so typically you only apply to, people usually apply to like less than 20 MD PhD programs. I think I applied to like 15. And then, MD PhD programs tend to be 8 years long. That's a really long time. The nice thing, though, is that your medical tuition is actually covered. So they pay you to go to med school, and that you also get a stipend that's similar to a PhD student stipend. So if you do MD PhD, don't go into debt for med school. And the way it typically works, you first do 2 years of med school, and this is normally like the classroom component of medical school. And then you do 4 years of PhD, which is on the shorter end of bio PhDs. Generally, if you're not in an MD PhD program, your PhD is going to take longer. And that's because they like get rid of some requirements that you have to fulfill to speed things along. And then you do 2 years of med school, which is kind of in the hospital portion. And then after that, you do residency. And after that, if you want to, you do fellowship. And typically the postdoc, if that's something you do, will be embedded within the fellowship, meaning that the fellowship / postdoc takes longer than either of those independently. But in theory, after you're done, a lot of doors are open to you. You can be a doctor, you can be a researcher, you can do both. And so you can, kind of the hope is that, at least my hope is that I will be able to do research on a disease and also be able to treat patients with that disease. And having knowledge in kind of both areas will allow me to really push a certain field forward. So I chose to do an MD PhD. Why did I do that? One is that I really love science and I can't really see myself not doing it in some capacity. And so the PhD was kind of a must because I wanted rigorous training in the sciences. I also like medicine. I really like talking to patients. I love learning about disease pathways. And the only way you can treat patients is by getting an MD. So that ended up also being non-negotiable. And ideally, I would want to combine them in the future, similarly to how some of my mentors did. And kind of a side note is that I would not go into debt for med school. And I also have some indecision, like I don't know whether I like science or medicine more, so why not just do both in the beginning and see what happens. And lastly, I thought it was a fun way to spend my 20s. This is obviously not the case for everyone, but as someone who likes science and likes medicine, like it's nice to get paid to do it and just kind of hang out with your friends while it happens. So I kind of felt like the lifestyle was a good suit for me too. So I ended up starting my MD PhD program at Hopkins. I didn't take any gap years, which is actually a little unusual. So that's where I am at now. Hopkins is located in Baltimore, Maryland. So I've come back to the East Coast, and yeah, it's a good hospital. It's a good program. This is me with some of my MD PhD classmates, and we're all just kind of living the dream. I mean, I say that having not started my PhD yet, though. So for now, I'm living the dream. And soon I will probably be very frustrated with whatever experiment isn't working. But yeah, that is where I am now. I still have a long way to go, but so far I have enjoyed myself. So I just want to leave you with some parting words. I know I went over time. I'm very sorry. I thought that I could finish this faster, but turns out no. These are my parting words. Ask politely for what you want. The worst someone can say is no, but no one is going to be able to say yes unless you ask. So just ask. It's fine. If you cast a wide enough net, you are going to get someone saying yes to you. And so just because you're getting rejected by some places or people doesn't mean that you should stop. If you just increase your denominator, you're bound to get some kind of signal. The other thing is that it's really important not to get too attached to any specific idea or lab or school, etc. You don't want your definition of success to be super narrow, and it can be really dangerous to get too kind of emotionally attached to one of these things because it kind of blinds you to contrary evidence. For example, you can join a lab because you're really interested in the science, and even though you don't get along with the PI, you continue to stick with it because you're really interested in the science, and then your PhD ends up being a really miserable experience. And this is actually a surprisingly common occurrence. So that brings me to choose a good project and an even better mentor. I would say if you are choosing between, a project you're really interested in and an okay mentor, and a project that you're like, you don't know that much about, but a really good mentor, I would go for the latter, because again, the mentor-mentee relationship is the wild card. The science is going to just happen. At the end of the day, like whether you're pipetting one thing versus another thing doesn't actually matter very much, but your relationship with your PI is going to change the trajectory of your entire career. When you're making career decisions, it's a good idea to talk to a lot of people and get their opinion. And importantly, you should try to collect both positive and negative experiences, because, you want to know the pros and cons of any particular path. So you want to be a good scientist about this, essentially. And I would not kind of like disregard negative experiences. Like, just because you find medical school a very attractive idea does not mean it's right for you. So you should be very keen to try to disprove your hypothesis. And if you find out that you can't, then you probably should go to med school. But until you have, figured out all the bad things that could happen to you if you did med school, you should probably not do it. And the last thing is that you should try to find joy in learning science and having fun with your colleagues, because science is really hard and academia is very kind of brutal, but you meet a lot of great people through it. And at the end of the day, you're working with very fascinating stuff. Like you're making real discoveries. And so it's important not to lose sight of that. That is my research journey.
Thank you so much.